Okay, good afternoon, everyone. It's an afternoon here in Nairobi. We've actually received some good morning greetings from the likes of Margaret. It's a lovely afternoon here in Nairobi. It's quite sunny, and welcome to our today's virtual talk that will be presented by Akbar Ganatra. And the title for today is on how agricultural pesticides promote Schistosoma host snail abundance by eliminating competition. My name is Richard Kipngeno. I work here in Nature Kenya as a budding and membership officer, and I will also be moderating today's talk. So just to remind all our participants that we kindly remain muted, so that we give our speaker ample time until he finishes presentation. And if you have any question, any concern, maybe any comment, you can always raise them through the chat box, and we will strive our level best to go through all of them when the presentation is over. Also, this to let you know that this presentation is recorded, and after we will upload it in our Nature Kenya YouTube channel. So you feel free to visit that site should you need maybe to rewatch it again. So ladies and gentlemen, just to introduce who our speaker is today, Akbar Ganatra is a Kenyan medical parasitologist and entomologist who is dedicated to mitigating the burden of parasite-driven diseases. He began his academic journey at Keele University in England, where he obtained his master's degree, combining his research interest in genetics with his newfound fascination with insects. His research focused on the genetic basis of speciation between molecular forms of Anopheles gambiae, a deadly mosquito species prevalent in his home country. In the year 2017, Akbar was awarded a DFG scholarship for his doctoral studies at the International Center of Insect Physiology and Ecology, ICIPE. His PhD research was a collaboration between the Kenya Medical Research Institute, as a KEMRI, and Helmholtz Center for Environmental Research, UFZ, in Germany. Akbar was registered at Egerton University and stationed in Mbita, Kenya, where he explored the impact of agrochemical pollution on the transmission of schistosomiasis in Western Kenya. During this time, he honed his skills and research interest in malacology, ecotoxicology, disease ecology, and freshwater ecology. Aside from his pursuits, Akbar has also been an active member of various scientific societies, which includes the East African Natural History Society, that's Nature Kenya, the Explorers Club Kenya, the British Ecological Society, BES, the Royal Society of Tropical Medicine and International Health, the International Society for Neglected Tropical Diseases, and the Global Schistosomiasis Alliance, GSA. His involvement in these organizations reflects his dedication to environmental conservation, global health, and research collaboration. Thank you very much, ladies and gentlemen, for listening to that in-depth introduction of our today's speaker. And allow me to welcome our speaker on stage. Welcome, Akbar, to take us through.
Thank you so much, Richard. So I'm not sure what's left to say anymore after that introduction, actually. So yeah, I'm Akbar, and yeah, I'm going to be presenting my PhD work. Yeah, I mean, again, I'll just get into it. Of course, when I need it, it has disappeared. So, hope everyone can see my screen.
Yes, we can see.
Yeah, so this is how the pesticides could be increasing the risk of schistosomiasis in Western Kenya. So to begin with, schistosomiasis is a neglected tropical disease. So this is a map of where it occurs. It's mostly in Africa. Around 90% of cases occur in Africa, but it also exists in South America and parts of Asia and Middle East. There are about 206 million people estimated to be currently infected, and the range of deaths are estimated to be between 17,000 and 280,000 annual deaths. Because it doesn't kill so quickly, the other way that we measure the impact of this disease is by disability adjusted life years. So this is how much a person cannot work because they're affected by the disease. And this is also quite high in 3.3 million disability adjusted life years. The figures, of course, kind of vary between who's doing the calculations. So they're quite large variability. Like I said, 17,000 to 200,000 deaths. We're not really sure. Yeah, it's quite morbid. You can see some of the pictures here. Yeah. So the biology of it, again, I have to apologize for this really complicated image. I don't have the rights for this simpler one, but I'm sure we'll get through it. And some people might not even know how the disease, affects you. So I think it would be useful. If we start with the eggs here, when the eggs that are released either in the urine or the feces of a person enter the fresh water, they hatch into miracidia. So if I can use the laser to show you where I am, number 2. Yeah, these hatch into miracidia. So these are a free living life form, and they have about 24 hours to live, where they search for snails of the right species to kind of infect. So they're kind of aligned here. So these Mansoni go for Pamphalaria snails, the Japonica go for Oncomelania, and the Hematobium go for Polinus snails. And basically, they hatch in the thousands, but not many of them survive. But even a single one of them surviving and infecting a snail, basically they go through asexual reproduction in the snail. So from one miracidia, they can now come out as thousands of Saccharine again. So because snails also can clone themselves, it's basically a single snail and a single miracidia in a water source can cause a whole place to become endemic. So the miracidia infect the snails, and then they go through this sporocyst successive generation. It takes like 2 to 6 weeks, and then the snail, it has its behavior altered as well. So it's like more advantageous for the parasite. They come more closer to the humans, and they start releasing the Saccharine daily. And this can go on for months as well. So like I said, a single miracidia can release thousands of these Saccharine. And these Saccharine now, they also have 24 hours to search for a human host. So they'll be in the water 24 to 48 hours. And then once they find a human host, they can actually just penetrate the skin. So they have digestive enzymes that they release. So you don't have to drink the water or anything. Like you can just have your foot inside the water and it can infect you. So like peak infection time is like if you've been in water, which it has these Saccharine and these infective snails, if you're in there for 30 minutes, then you're like probably guaranteed to be infected. So once they penetrate and they enter you, now it loses its tail and becomes something called a schistosoma, and it uses your blood circulation system. So first it moves into your liver, and then it matures into an adult. So this also takes like about 6 weeks or something. And then they then move into now their preferred area depending on the species. So the Hematobium move to your urinary bladder, Mansoni move to your intestines, and the Japonicum can do either actually. So now when these adults, they have to kind of be in a mating pair. So actually you have to be infected with 2 of them, a male and a female. And then they kind of lie in copula. So the female lies within the male. He's kind of like shaped like a pocket, and she just lives with that inside him. And then they go on drinking your blood from wherever they are, either your bladder or your intestines, the capillaries there. And then they use that nutrients to lay eggs, so that those eggs are now released with your stool or urine back into into the system. So I'm just going to dwell on this part for a minute, because again, I'm assuming a lot of people don't know about the disease. So basically what happens is these adults, they do want to release their eggs into the wild, but the eggs are just so small, they get reabsorbed into the bloodstream. And what happens is they're in circulation, and because your body has like, filtration systems to get out all these things from your blood, they get stuck in those places. So like your liver and your spleen, they get concentrated with eggs, and your body doesn't really know what to do with them. So it just covers them with scar tissue. So that's what happens. You have like the enlarged liver, as you saw in the first picture. That's just like a liver that's been clogged with eggs and covered in scar tissue. And this happens in almost any organ. It's just really likely in the liver. But basically where the eggs get clogged is what causes the disease. So you can have like, hepatoschistosomiasis. If it gets stuck in your brain, then you have cerebro or neuroschistosomiasis. So it can cause really severe problems, actually. In the case of cerebro or neuroschistosomiasis, it can cause strokes, for example, so and paralysis as well. Yeah, so on that morbid note, to the next problem. So agrochemicals, there's an ongoing debate on what should we actually do to feed the world. We need pesticides, but they're killing us, but like we have a growing population and there's not enough food. So while we apparently need pesticides on one side, the other side is like pesticides are more harmful than we think, because there's like the obvious harm of like, non-target species dying. But over here, someone has tried to make it clear by the different impacts. So yeah, loss of natural enemies to pests. Oh, Islam Alisa, yeah. So the pesticide resistance is another thing. Even though we're using pesticides right now, the cost of resistance is increasing. It affects other areas of public health. For example, this schistosomiasis that I'm going to bring, crop losses from basically other plants also being affected. It affects domestic and animal wildlife. So it affects groundwater, it affects bird loss, fisheries. So there's actually a widespread impact of agrochemicals that we need to actually address. So in Germany, they're a bit more conscious than we are, and they were developing a new kind of method of assessing the impact of these pesticides. So they already do chemical analysis, but of course that's very expensive and it's very timely. It can take a really long time. I'll explain to you how long it took in this project as well. Yeah, so they were coming up with a bioindicator system. So you can see it's called SPEAR, species at risk. And essentially is if you choose a 50-meter kind of section of a river like this, and you do sample collection for these macroinvertebrates. So you just collect every insect that you see inside this river. And then based on what you see, because some organisms are more sensitive than others. So if you see the sensitive organisms, you can actually estimate that it's not that polluted. But if you only see the tolerant ones, then you can say, okay, this has an amount of pollution. So based on how many of these you can see, they can actually make a really good estimate of what the pollution level is. I think there's some of this in this presentation too. So we tried to do this in Kenya, but the point is, while they were doing this, they had to kind of assess the sensitivity of each macroinvertebrate to see where it sits on the scale. And so while they were doing this, they came up with this scale, and that's when they noticed that Planobidae, this is like the host snails of schistosomiasis, they are the second most resistant to almost any kind of pollution. So this is when the hypothesis came, okay, if these snails are more resistant to pollution than everything else in the water, so if the water is polluted, then the only things that will survive are, these hosts of a disease, when the disease go up. So that's how the hypothesis was made. Just for reference, that's the most resistant to pollution, this Eulemellibranchia. It's a mussel. So yeah, so this was my experiment now to find out if this was the case. Are agrochemicals actually going to increase the risk of schistosomiasis? So we selected this area of Kenya because it's highly agriculturally relevant, and it's also got schistosomiasis. So yeah, we have Homa Bay, Kisumu, Kericho, and Migori. So these were because they have different kind of crops as well. So we assume they have different cocktails of the agrochemicals. So here we had mostly subsistence maize farms. Down here we had sugarcane plantations, we had rice plantations and tea farms. And as you can see, this is, well, no longer recent, but this is like the presence of the disease in the region. So this was 2014, that was 3 years before I started my research. So we could estimate that the disease was still highly, present in the area. If you're a little bit more interested in this, I can send the paper over. But my main concern is the disease is present almost in the entire region. So I selected sites from all the regions present, basically again spread out over the rice plantations, tea plantations, sugarcane, and also trying to select sites where I thought the snails might be present. So this is me collecting the snails, or basically anything I find in the water, trying to divide them up. This is an example of what you saw. So kind of a lot of mayflies, some damselflies, one small snail here, some beetles. So this is kind of clean water, you can estimate. Then basically we did lab analysis. Oh, I think I'm missing a photo. We also collected water samples. So these water samples were sent to my friend Faith here, who's also in the chat. You can say hello. She did the chemical analysis on the water. So this is to find out the exact pollutants, the exact chemical composition of the water. Basically, we saw everything in it. And then we did two things. So the first is I did experiments on the macroinvertebrates of the area to see the different sensitivities, similar to what they did in Germany, just to see if our macroinvertebrates had similar sensitivity to the German ones, and to see if our hypothesis had any value. And then the other thing is basically each site had like a different kind of cocktail of agrochemicals, and to relate each site, you had to convert it into a standardized unit. So we call this a TU max. Here's the formula for it. It's basically just like the relative sensitivity of an organism to each compound, and then we compare that by site. It's just to tell us like how much the pollution is in one site compared to another site, regardless of what's in it. So here's the results of my experiments. So this was again just to confirm that we had the right hypothesis. So over here you can see that the host snails are actually the most tolerant. They were actually so tolerant for this compound, imidacloprid, which is a neonicotinoid, that we couldn't actually plot it on the graph. So over here you can see every other macroinvertebrate had like a sensitivity lower than the ones for the snails. So as high as the pesticides go in that river, the snails will not die, but everything which is either a competitor or a predator could possibly be affected or will probably die. And this is the case for diazinon as well. But we could actually kill it, but you can see it's still the highest in the graph. So it took the most concentration. Now on this one is basically now where the TU comes in. So on this scale, -5 means almost non-polluted. There's no trace of pollution, and 0 would mean very polluted, or basically it would mean if you put a sample of the reference organism in that water, half of them would die in 24 hours. So it's a logarithmic scale. So -1 is 1/10 of 0, and -2 is 1/10 of -1. So -3 is what we say is moderately polluted. So here we have no pollution to moderately polluted. That's when the snails start to appear, when the water is at least moderately polluted or greater. So that was our first bit of evidence. We don't see the snails in like clean water. We only see it where the pollution starts. Then we tried to test the effects of the independent effects of the different things. So as you, I didn't mention so much, but yeah, we collected the physical chemical parameters of the water. We had 35 different variables to see where the snail presence was. So these were the ones that had the most effect on the presence of the snails. So this is the dominance of the competitors, the pesticide pollution. So this is basically what determined where the snails were. Like if there was pesticides, if the water was very turbid, if there was like, less dissolved oxygen or high number of species. And then these variables we took for further investigation. So we put them into a PCA, which is a principal component analysis. So these pesticides we can see kind of correlate with turbidity. So this is kind of an indication that our calculations are correct. So pesticides and turbidity is always correlated because turbidity increases when the amount of rainfall in the water increases. The amount of rainfall increases the amount of water in the rivers. So the turbidity goes up, and so we expect pesticides to go up. So the fact that we see these two correlating is a good idea, for a good suggestion for us. The same with O2 and species richness. So the higher the number of species, the less oxygen in the water because it's being used. So over here, as pesticides increase, the number of competitors of the snails seems to be decreasing. So this is how we came up with the conclusion that the pesticides, when they increase, the number of competitors of the snails decrease, and this is what's driving up the number of the abundance of snails. Over here, we compared now the community composition between low, medium, and high pollution. So essentially, the community composition is like the trophic pyramid, like the number of grazers and the number of like predators you have of those grazers. So what we see is actually there's not that much change in this community composition. That in that sense, the number of predators stays kind of the same. But what we're seeing is in the grazers, which is this part here, the dominance of the snails is increasing. So actually what you're having is the competitors are reducing and just being replaced with the tolerant snails. So essentially you have, with more pollution, more snails. So yeah, that's basically the talk. The conclusions are that the Schistosoma host snails are more tolerant to organophosphates and neonicotinoids. So those are the experiments I did in the laboratory. The Schistosoma host snails were only found in moderately polluted waters or greater. So they're not found in clean waters. They seem to only like the polluted waters. And the community composition, while not being altered too much by pesticide pollution, the grazer gill itself is quite altered with more, it's being dominated by the snails. So the risk of disease increases because of this dominance of snails in greater pollution. And the study shows the effect of agrochemicals actually begin at lower concentrations. So we don't need things to start dying, but like, the overall community is affected. So that's already a problem. Next steps. So better regulation, I would say, into pesticides in Kenya, and remediation. So there's methods of how they have sanitation plans for sewage. They've made something like this for pesticides, so we can do that. New technologies so that we don't have to actually use the pesticides. So there's like push-pull, which ECP works on, maybe GMOs. And for schistosomiasis, we need to employ WASH, so water sanitation and health programs, alongside mass drug administration. That's the talk. That's the paper. If you want to download it, you can scan that right now, and I'll welcome any questions.
Thank you. Thank you very much, Ganatra, for that insightful study and for sharing your findings with us. So I will open the floor to the participants. If there's anyone with a question that would like to raise to the speaker, feel free to do so. You can raise up your hand and we'll give you an opportunity to do that. Meanwhile, you can also raise them through the chat box, and we will sample each and every as we go along. Margaret, you have a question?
I was going to ask, so if you have a snail, does that mean every snail has got the schistosomiasis? Or do you have some snails that are free of this pest? And the other question is, do you have a natural predator? Because usually if you have one type of organism, you usually have a natural predator as well. And how do you increase that?
Okay, so in the case of how infected they are, I would say in the snails, the prevalence is actually quite low of this particular parasite, but snails generally tend to always host a parasite, I think, of some kind. They just happen to have not, they don't have like a skeleton, like an outer skeleton or something like that. They just like squishy, gooey food for everything. So they'll always get infected. But the rate of finding schistosoma in a snail, you have to find the right snail. And then in that case, it's also still around 1%. So you're not going to have many snails being infected by this particular parasite. But like, I think it also depends on the site you go to, because we had some sites with up to 17%. So of the snails we caught having the infection. Yes, Charles? I don't know if your question is related to this one, because she still had one more question.
I did. I was just asking about natural organisms. Usually if you have one type of organism, then you usually have a natural predator. So is that?
Yeah, so basically, actually, again, like I said, the snails are just squishy, gooey food. So almost every insect we come across, if it's carnivorous, it can feed on the snail. But we do have specialist predators as well. I didn't come across them in my work, but there are some a specialist parasitic fly that affects these snails. There's a different type of snail that eats the eggs of the other snails as well. So that's a predator, we consider it. But yeah, in our study, basically, if it was an insect that ate, if it was a predatory insect, we consider it a predator of the snail, because just generally, a snail is a very easy meal. They're eaten by almost everything. Sorry, yes, Charles? You're on mute, by the way. You're muted.
Okay, thank you. Okay, there's someone with a question?
Yeah, sorry. I put a couple of things on the chat, but I'm just wondering, apart from that, which is really interesting, I thoroughly enjoyed your talk, how are the authorities responding to this? Because quite clearly, if you don't get the support from the higher levels, the agricultural departments, the Ministry of Health or whoever, it's interesting work, but it needs to go further.
Yeah. So at some point during my PhD, they did come to do a public forum on the effect of pesticides, and I did give them this information. And one of the pesticides that I've researched on was banned. I wouldn't say directly because of this research, but yeah, they did ban them. But then the farmers are currently appealing the ban as well. So it's not just the government. It's like we have to, raise awareness throughout the whole, stakeholder system, because the farmers also need to have a replacement for the pesticides, because they're for the poorest of the poor, they don't really have time to wait for science to be wait 2 years and it'll give you the answer. They're dying, their crops are dying right now. So it's unfortunate, but there are other methods of plant protection, but that also need government assistance. And so my kind of strategy right now has been to focus on the disease, and I've also given some recommendations for that. So we'll see how that pans out. Yeah.
Oh, well done. Very good.
Thank you very much, Charles, for that question, and also to Akbar for answering it. Is there any other one with any other person with a question?
There's a question about any recommendations, but I hope my last slide answers that, or I can go into greater detail if anyone wants.
Oh, I just had one question, and that was, is this similar with bilharzia?
Yeah, so bilharzia is kind of when the disease was named after the person who discovered the life cycle, but then they decided to kind of stop naming diseases after people. And so we're like trying to shift the name to Schistosomiasis, which is named after the parasite.
Okay, so when we were, I mean, I grew up in Kenya, and I always remember we were told not to go into still water, and that the snails were usually found in still water rather than running water. So is that true or not?
Yeah, that's a little bit true. But like, I mean, we found slow moving water. I mean, we still found like the highest number of the snails in our study in like a river, where it was like a slow moving river. That's still a little bit deep. But yeah, I mean, the sites kind of varied. We had like ponds and, dams with the snails, and kind of, it has, as long as it's fresh water and as long as it's not so fast moving, they can kind of colonize it. Yeah.
Okay, thank you for that. I see Geoffrey Ongondo has raised his hand. If you have a question, feel free to unmute and ask.
Thank you for the talk. And I work as an aquatic ecologist, and one of the things that I think I probably would like to know is, did you work on the incidences of disease? Do you have any information in terms of, at the moment, what is the current incidence of schistosomiasis in the population where you carried out the research?
In the population of humans?
Yes.
Not really. I mean, for some of my experiments, I did have to like sample some children in my specific town so that I'll be able to, continue doing those research. But like that happened to be really high. In one school, we went to like 66% of the children we tested had bilharzia. Or 66, I think, was the average. One place was 70 something and another place was 40 something. So yeah, but not in every place we went to. And even when we were collecting snails, because I was actually mostly working on the incidents in the snails, because actually my PhD was supposed to be for 3 years, and getting ethical clearance to work with humans was, we foresaw that going to be taking too long, so we didn't even consider it. But yeah, in the snails, we only had one site with infected snails, but even that, it was like quite high. I think for Bulinus, it was like, which is the Schistosoma hematobium, that snail, 17% of like those snails had the infection, which is, I thought, quite high. And like in terms of prevalence, we were told to expect about 1% of the snails.
Okay, so thank you, because based on the fact that this is one of the neglected diseases, then I believe it could be still a very big problem in part of these areas where the snails are found. And that is why I asked if you had worked on the human prevalence also.
No, actually, I can go back to that image because essentially that work had already been done. Like I said, I don't know if you guys can still see. Yeah. If you go to this image was done like just 3 years before I started my PhD. So it was essentially like the incidence in the area in the humans. You can see 50 to 100% where it's red, 10 to 49% where it's brown, and yellow is up to 10%. So green is where it's not, but you can see there's like patches of red almost, yeah, in here. So we didn't really feel a need to redo this work, because another thing in science is like, is your research novel? Is it new? Or are you just repeating what someone else has done? So there's no need to kind of look at the prevalence in the region. We already have that information very recently. It was less than 5 years by the time I started my research. But yeah, I already like where I did it was this area, and some studies found prevalence to be 100% in children. Some studies 70%. We found it to be 60%, but we still went and gave that information to the local authorities, and we gave them the medicine and told them, go treat these people. Well, we give the ones we found infected, but the government needs to go and treat the entire school, we feel, because that's quite a high prevalence rate.
Okay, thank you for your answer.
Thank you for that. Any other person with a question to the speaker? Maybe additional comments?
Yeah. Just my acknowledgements of everyone who helped.
Oh, great. It's nice. Okay, is there any other person with a question before we end the session? Any comment? Any compliment?
Yes, I'm Fleur Ngueno, and I'd like to say thank you very much. That was really interesting. And greetings to Charles Dewhurst, an entomologist who has been with Nature Kenya for many, many, many years. I hope that all this research will actually move forward to help people and reduce pesticides.
Thank you very much, Fleur. We really appreciate it. Thank you.
Thanks, Fleur.
Yeah, thank you. And the disease, I hope you're all aware of the disease now and maybe a little bit how to mitigate it.
Thank you very much for that. And maybe also, Akbar, you can share maybe your email address in the chat box so that maybe if the participants would want to follow up for more information, they can easily reach out for you.
Yeah, sure.
Okay. As he shares his contacts, thank you very much, everyone, for creating time to join us in our today's virtual talk. From this side is to wish you a happy Ramadan holiday ahead of us and a happy weekend. Thank you very much and have a nice time.
Thank you everyone for joining.
Thank you for being such a good host.
Thank you.